We are now moving on to Unit 4 for the exam review. So Unit 4, if we remember, is all about cell signaling and how cells talk to each other. So, things you should know. There are 3 major components of any cell communication. We're going to talk about the different types and how that works, and a couple of examples of each. And then we're also going to talk about how we then trigger certain things of normal functioning via communication. So, how do cells signal with each other? It's possible that they are directly in contact with one another, whether it's gap junctions or in plants we learned about plasmodesmata. There is secreting local signalings. So this includes things like neurotransmitters being released from one neuron to the next neuron, or there might be growth factors that are released into the surrounding environment for the cells right around it. And then there's long distance signaling, which is more for hormones that might be, released from the pituitary and travel to all different parts of the body in the bloodstream. The 3 major steps of any cell signaling pathway include reception. So you have to receive the signal, and this is kind of the detection of some kind of signaling molecule. And we call that signaling molecule, whatever it is, a ligand. And it's coming from outside the cell somewhere, whether it's direct contact with another cell or it came as a hormone signal or whatever it may be. Some kind of ligand is going to come from outside of the cell, and a cell needs to receive that message. Transduction is somehow able to transduce the signal. So it will take the ligand receiving response and then convert it to some kind of downstream response, downstream form. Maybe there's a conformational change that occurs or an activation of a particular protein that will then trigger a downstream response, which is the 3rd part of cell signaling, the response. Whatever the reaction is to that initial signaling molecule. And because it talks about outside of the cell and then responses inside, the plasma membrane plays a huge role in all of this. So here's kind of that 3-step process. The signaling molecule in this case is red, so the ligand would bind to an integral protein here in the membrane that's acting as a receptor. And ligands are very specific to their receptor molecules. So in this case, kind of like the lock and key portion, there might be some kind of conformational change or activation of some relay molecules, as they're called. So it's transducing the signal, and then that leads to whatever activation or turning on or response that's taking place inside the cell. So here's kind of rehashing that reception part. So a ligand binds to a receptor. As I mentioned, it's highly, highly specific. And it can either be a plasma membrane receptor, like in the picture previously, or it's possible that it's an intracellular receptor, meaning that there are molecules inside the cytoplasm or the nucleus that can act as a receptor. Those will only be receptors for small or hydrophobic ligands, the ones that can cross through the membrane on their own without any assistance. So if you remember all the way back to the plasma membrane, hydrophobic molecules can go freely through, or very small polar molecules can go freely through the membrane. So if those can come in, then they can actually bind to receptors inside the cell. Otherwise, the ones that are hydrophilic or water soluble are the ones that have to bind to the plasma membrane. So they're going to bind to a receptor that's located there. And then there's usually some kind of conformational change, and that is what triggers the transduction signal. One that we learned specifically about is the G protein coupled receptor. So this has that 7 transmembrane kind of protein embedded in the membrane. And then on the bottom here is where it would interact with a G protein. and then the ligand would bind right here on the top to the binding site. So it would bind and then cause a conformational change that would then activate a G protein that's on the interior of the membrane. So here's roughly how that works. So the signaling molecule binds. The activated G protein, due to conformational change, is now freely able to, for example, come over here and activate a enzyme that wasn't active before. So this GTP molecule can kind of come over here, activate, and then cause the intracellular response. So again, that's a G protein coupled receptor because it involves the activation of a G protein that can then turn on things downstream. The other type that we learned about is ligand-gated ion channels. So the ligand binds to a receptor just like it did before in G protein coupled, but instead of having a G protein, it's really a gate. So the ligand binding causes a conformational change that would then open the gate and then allow things to go through. So maybe it's a pore of some kind for specific ions that wouldn't normally be able to go through on their own. And then once those are inside the cell, they can trigger some kind of cellular response. And when the ligand leaves, then that channel closes once again. Kind of circling back to intracellular receptors, they are found in the cytoplasm or nucleus. The signaling molecule must pass through. And oftentimes they end up controlling gene expression. So because they're inside the cell, they can control, as we learn later on, which genes might get turned on or off. Here's kind of a separation of the three different kinds. On the left, you have the G protein coupled ones. On the right, the ligand ones. And in the middle are the intracellular ones. So once the message is received, then it needs to be transduced. So it has to somehow be triggered into a form that can bring about a response. This is oftentimes where you see something called a phosphorylation cascade. So cascade just implies that there's lots of different steps between the initial molecule activation and the target molecule activation. And it usually involves a series of phosphorylations. Phosphorylations usually activate things. So the enzymes that actually phosphorylate are called kinases. So there might be a lot of kinases that just kind of come along and phosphorylate, and then at the end you get the response that you're looking for. So here's a little bit about a picture of what that might look like. You can see that there is energy involved. So the idea of phosphorylating oftentimes requires energy to take place or to be used in the production of that cellular response. Sometimes there's also second messengers. So a second messenger is where something in the cascade can act as a ligand for a different receptor. So cyclic AMP or CAMP is one example of a second messenger. Calcium is another, IP3 is another. But the idea here, in the picture below, it's IP3. You have your initial signaling molecule. The first messenger is the ligand, and it binds to the G protein coupled receptor, and you get some activation, and you create IP3. Now that is called a second messenger because it can then travel to, let's say, the endoplasmic reticulum, and it acts as a ligand to a calcium channel. So it's called a second messenger because it wasn't the first, but it can trigger a separate cellular response downstream. The third is the response part, and that just means the result of the action. Most of the time, it has to do with gene expression. So a lot of times it might be turning a gene on or off, or it might be enhancing or inhibiting a specific protein that's supposed to be functioning in the cytoplasm for a more immediate response. The ones that control gene expression are more for delayed, longer lasting response. If you need immediate action, then it's more for regulation of proteins in the cytoplasm, but they don't last as long. So apoptosis is thrown in here too because it does have to do with cell signaling. Apoptosis just means that it's triggered cell suicide. So there's a series of enzymes that basically do this. It is a cascade of caspases that is triggered in some way. So how does this happen? There might be signals released by surrounding neighbors that say, hey, the cell is damaged, it needs to be taken care of. Apoptosis also occurs naturally in animal development. Think about, webbed fingers turning into regular fingers, those kinds of things. So there are traditional cell signaling cascades that do that too. All right, so then homeostasis and regulation. Within signaling, there's a lot of control happening to kind of maintain our homeostasis, which we need in order to stay alive. So our endocrine system is the system that does all of the hormones. So the long distance signaling can trigger chemical signals for slow acting but long lasting effects. Whereas the nervous system is more local signaling. Those do very fast responses to things like other neurons or muscles or even endocrine cells to maybe then release hormones. So there's coordination involved between the different systems to try to maintain chemical levels and heart rate and breathing rate and temperature and all of those things, which is the idea of homeostasis. We have a set point for a lot of things. So if we go or deviate beyond that set point in either direction, that can act as a stimulus. There's some kind of signal there, and it can then be received by the sensor, which would in this case be a receptor of some kind, and then trigger a response. So it really is kind of a signaling cascade type thing. In terms of maintaining homeostasis, we can see that there might be positive feedback or negative feedback. Negative feedback is way more common. It's kind of like a thermostat. If we go beyond, then we turn something off. Like if we get too hot, then we might turn off the heater. But then when we get too cold, we might turn on the heater. So it kind of bounces back and forth to maintain glucose, temperature, pH, other things. Positive feedback is where you continue to amplify the signal. It's more rare because we oftentimes don't want to make more than we need at any given time in a cell, but it does happen sometimes, like ripening of fruit, the onset of labor and lactation in mammals, those kinds of things. Certain animals maintain homeostasis in different ways. There are endotherms, which are the warm-blooded animals that generate heat by metabolism. So they need to maintain a proper internal temperature. There are also ectothermic animals. These are the quote-unquote cold-blooded ones. And so they need to get heat from an outside source, like the sun and a heat rock. We talked previously about metabolism, which is the total amount of energy an animal uses for a given unit of time. So ectotherms, because they can't really easily maintain an internal state, they have to have a lower metabolic rate. It's also inversely related to body size. So thinking about how super small animals have a high metabolic rate and very large animals have a small metabolic rate. And so we've adapted many, many different things to try to maintain elements of homeostasis, such as thermal regulation. All right, so now I'm going to talk a little bit about the different systems just to kind of give you a background in signaling. In terms of the nervous system, it's similar to the three-step cell signaling process. There's some kind of sensory input that is received. It is integrated by the brain in some way, and then you get a response, or in this case, a motor output. And those take place in different elements of the nervous system. The primary unit of the nervous system is called a neuron. So that's a nervous system cell. And it contains the cell body, which is the main part in the middle. It has dendrites, where information is coming in, and then information travels down the axon to the axon terminal, where it will connect with the next neuron across a tiny space called a synapse. So chemical messages, in this case neurotransmitters, can get released by one, go across the synapse into the other. And that's really how the traveling of information takes place from one neuron to another. And so this slide basically covers all of those things. You can take a minute to pause and read through them, just kind of knowing the hallmark components. Now, in terms of cell signaling, there is something called a membrane potential. So neurons are controlled by ion-gated ligand-gated ion channels. There are sodium channels and there are potassium channels. There's also a sodium-potassium pump that basically does active transport to maintain certain levels of sodium-potassium across the membrane. So there's something called a membrane potential, which if you added up all the positive and negative charges, there is a difference across the membrane. So it has the potential, if you start opening channels, to have an influx of charge. And again, the pump is responsible for maintaining the negative potential inside the neuron. So at resting state, things want to basically rush in. There's a negative charge on the inside of the cell, and the pump actively maintains that. When there's an impulse that actually goes, that's called an action potential. And what that basically does is opens up, they are called voltage-gated channels. The change in voltage causes those channels to open. It acts as a stimulus. And once those open, then sodium can rush in because it's going from an area of high to low concentration. It's going to enter the cell and then cause a response. From there, potassium channels also open and try to compensate, so potassium leaves the cell. And that combination of things basically triggers what we call an action potential. And that triggers the signal down the axon. So here's kind of what that looks like over time. You can see it's normally a negative charge, and then there's a slight increase in voltage. There's a magical threshold where once you cross it, then it really does kind of open up the floodgates, and there's a huge action potential. And then the pump tries to pump everything back to where it was. It's sometimes called undershoots, where it kind of goes too negative, and then it fixes itself and maintains back to baseline. So that's the super quick version of an action potential, but if it's a bit confusing, please go back and look at the detailed version for this particular chapter. Neurotransmitters are released at synapses. So as I mentioned, they are in vesicles, and those vesicles get released at the membrane of the presynaptic cell, and then they travel across to receptors. They act as ligands to receptors on the receiving or postsynaptic membrane, which can then trigger that action potential in the next neuron. Neurotransmitters can be excitatory or inhibitory. So depending on what it is, it can actually excite neurons, or it can turn them off. So depending on if you're turning them on or turning them off with specific ligands or specific neurotransmitters, you can get different brain cellular responses. Okay, so quick rundown of the endocrine system. As I mentioned, traditionally endocrine has to do with long distance signaling in the bloodstream. Paracrine is really for just in the surrounding immediate area. So there might be a release of hormones or signals that act as ligands to surrounding tissues right nearby. There's even something called autocrine signaling, where a cell can release something and it triggers a response on the own cell. Those are a couple different instances where hormones might come into play, which is located here in the middle. Local regulators are those examples of adjacent cells or local signaling. And on the left, there's also something called pheromones that we didn't really talk about, but thinking about the sense of smell, certain organisms release kind of smell pheromones that then trigger responses. We've already talked a little bit about negative feedback and positive feedback, and this is just one example of how that can occur in an endocrine system. Here's an example of the positive feedback, in this case with lactation in an endocrine system. When we're talking about hormones, there's two different types. There's peptide hormones and steroid hormones. Peptide hormones are water-soluble, meaning that they are hydrophilic or polar. Because of this, they go through binding of receptors on the plasma membrane. So this is where your G protein coupled receptors come into play. They generally, because they go through the membrane and signal a transduction pathway, they are usually just changing protein activity that's already in the cell, in the cytoplasm. So because of it, it's a really quick response, but it's short-lived. It only works on the proteins that are in the cytoplasm at the time. So that includes things like oxytocin, insulin, epinephrine. Steroid hormones, on the other hand, are hydrophobic. They are lipid soluble. Those are the ones that can just enter through the membrane on their own. They don't need to go through a membrane protein. So they act on intracellular receptors. And the intracellular receptors are really the ones that change gene expression. So they turn genes on or off. So because of that, there's a lag time. So it's a little bit slower, but because you're changing gene expression, it has a longer life in terms of the response. So these are traditional hormones like testosterone, estrogen, and so on. So here's just kind of showing you how those two are different in terms of cellular response, with peptide on the left and steroid on the right. Plants have their own responses to different things. So for example, one major plant hormone is called oxen, and it stimulates cell elongation. So if this hormone is triggered, I know the font's a little messed up here. If this particular signaling pathway is triggered, it will cause cells on the dark side of a stem or plant to elongate faster. And because of that, the ones on the light side won't elongate, and it will cause the plant to actually shift and turn towards the light and exhibit something called phototaxis, where a plant kind of shifts towards the light. And that's in response to the chemical signal oxen. There are other hormones as well that plants use. One common one to easily think about too is ethylene. That's the one responsible for fruit ripening, which is an example of positive feedback. There's some responsible for cell division and so on and so forth. In terms of plant movement, I just mentioned phototropism, meaning that they bend toward the light. The roots of the plant actually exhibit negative phototropism because they're growing into the ground away from the light. But plants also respond to gravity, and some of them respond to touch. We also have something called turgor movement, which if you think about turgor pressure, if you have like a limp, flaccid plant and you water it, then it can, plump up and stand up. They have many responses to stress as well. So depending on the environmental conditions, plants have adapted ways that they can kind of respond. So if it is drought, they might close their stomata or do a couple other things, or if they don't have enough oxygen or so on. So you could always pause to kind of see some of the adaptations that plants have acquired over time. And all of these are a result of specific cell signaling and proteins and so on. Okay, touching on the immune system, I don't think you're really going to have to go in crazy detail. Just know that innate immunity is not specific. It is the one that you are born with. All plants and animals have an innate immunity to certain things. Whereas adaptive immunity is pathogen specific. Only vertebrates have this, and it's kind of our acquired immune response. If you think about having to, be infected with the disease to then not get it again, that's the adaptive piece. Innate, we have things like our skin, our mucous membranes. We have specific cells within us that just exist. Whereas the adaptive one are specifically antibodies and other cells that result from initial exposure to a pathogen. Inflammatory response is just one particular example of a signaling cascade. So if you were, hit with a splinter that had a pathogen on it, there would be all these kinds of signaling molecules released by specific immune cells, and those signaling molecules can then trigger all kinds of signaling cascades and cause different cellular responses as a result. In this case, phagocytes are coming and they are engulfing and eating all of the pathogens. An antigen is one of the cell surface markers that can trigger a response. So it acts as a, in this case, the ligand on the surface of the pathogen is an antigen. It basically says what type of pathogen it is. And then your cells have receptors all on the surface. So if they come in contact with one another, then that's one of the cell signaling responses, where the antigen is the ligand, there's a receptor on the immune cells that can trigger a response. There are different kinds of immune response cells, but I don't think you really need to know the specifics. In terms of active immunity, those are part of our acquired immune system, where we have memory cells that kind of remember what we've been infected with before, and therefore can defeat new pathogens if we encounter those same type again. We have immunizations and vaccines, which help introduce us to the memory of a disease without necessarily having had to have the disease before. And then there is something called passive immunity, where a mother's antibodies can actually pass to a baby. But in the grand scheme of thinking about cell signaling, again, these are antigens that are acting as ligands, and the immune cells are acting as receptors. And when those receptors are triggered, then that can introduce an immune response. The last part of this particular unit has to do with the cell cycle and regulation and how things can go wrong and cause cancer. So most of this should be a review from freshman bio, so hopefully this part's not too bad. So the life cycle of a cell is basically just called the cell cycle. And it's entire life up to the point where it divides. So why do we do this? Reproduction, growth, and tissue repair. Continuing with the vocabulary, our genome is just all of the cell's genetic information. So it's basically all of the DNA that one cell contains that needs to then make its way into every future cell or daughter cell after division. If you remember, prokaryotes have kind of one singular circular chromosome floating around in the cytosol, whereas eukaryotes have linear chromosomes. And different eukaryotes have different numbers. So humans, we have 23 pairs or 46 total chromosomes, whereas a mouse has a total of 40, fruit flies have 8, and so on and so forth. So they are organized in linear pairs, and they are found inside the nucleus of a eukaryote. So the idea is, before a cell divides, you have to first copy the DNA. And this takes place during one portion of the cell cycle, which I'll talk about in a minute. But the vocab can be a little tricky here. So in this case, one chromosome becomes duplicated and it's still stuck together. It looks a bit like an X here, and it's attached by a centromere. Those are called sister chromatids. So it might be chromosome 1 with its copy of chromosome 1, its duplicated chromosome. For right now, since they're still attached, this is considered one chromosome and two sister chromatids. Once they separate, they become their own chromosomes. Inside of your cells, you have the traditional somatic cells. These are your body cells. They have 46 total chromosomes, and they divide by the process of mitosis, which we're going to talk about here. They are diploid. They have two sets of chromosomes. There's two chromosome 1s, two chromosome 2s. So diploid or diploid literally means two sets. Our gametes are our sex cells. These are the eggs and the sperm. So we're going to come into more detail with this when we future talk about meiosis, which is how they divide. But they only have one set, which makes sense because sperm and egg, when they come together, will then have the two complete sets. So for humans, they have 23. So that after the egg and the sperm come together, you end up with 46. This is what the cell cycle looks like. You can see that 90% of it is in something called interphase. So this is the non-divisionary section, and it's broken out into G1 for growth, S, which is where the synthesis of DNA occurs, and G2. Then all of mitosis, and then the actual division of the cytoplasm, cytokinesis, take place in this M phase, which is just a short portion of the cell's life cycle. And then this repeats over and over. So in interphase, during the growth part, it's really just growing and carrying out its normal daily life functions. And then, as I mentioned, S stands for synthesis. So in the S phase, that's where those chromosomes actually get copied. G2 is getting ready for cell division, so that by the time it gets to M phase, it can actually do the cell division that it was planning on. I always remember mitosis by PMAT, which stands for prophase, metaphase, anaphase, telophase. I don't think you're going to have to go into any kind of intricacies of the different steps, but just knowing that kind of prophase is when the chromosomes condense outside of just a spaghetti-like chromatin. They condense into chromosomes, the nuclear envelope disappears. The microtubules start to attach to the chromosomes and move them around. By the time you get to metaphase, meta means middle, all of the chromosomes are nicely lined up in the center, so that in anaphase they split apart. So that kind of ensures that you have one set of chromosomes moving towards one side and the other set of chromosomes moving toward the other side. And we all know what happens when there's problems with that. And then telophase is when the things start to kind of reform in cytokinesis. Eventually they split into two cells. And I think if you just know that level of detail, you should be more than fine. There are a number of checkpoints along the way just to kind of quality control and ensure everything is going well. If there aren't any checkpoints, then the likelihood of having messed up cells goes up, and you can end up with cancer, for example. So there's a G1 checkpoint. There's one at G2 right before M phase, and there's one during the process of mitosis. And basically, they do a quality control and say, okay, you can keep going, or they say, no, it needs to stop. The G1 checkpoint's job is basically to say, okay, is the cell big enough? Does it have enough growth factors? Is the environment ready? This is the most important checkpoint, because if it passes this checkpoint, then the cell is going to go ahead and divide. If not, then it enters what's called a G0 phase, which is basically a pause. It's kind of a non-dividing state where the cell can just continue to live its happy little life. Certain cells in our body live most of the time in G0 and maybe never come back. Some cells can continue to divide after, but it's basically a pause state. G2 is really a quality control for DNA replication. So right before G2 is the synthesis phase. So when it gets to the G2 checkpoint, it's checking for any possible mutations, because if that's true, then the cell shouldn't continue to split with mutated DNA. The M phase checkpoint is basically just looking to see that all of the chromosomes are lined up in the center correctly, so that when they do split for anaphase, that you get an equal number of the right chromosomes going to the right sides of the cell. Externally, you can also have control factors as well. If there are growth factors released, thinking of overall cell signaling, it might promote cell division. There's something called density-dependent inhibition, where when the cells start bumping into each other, they naturally want to stop dividing. This is another direct contact cell signaling kind of ligand receptor type situation. And then there's also something called anchorage dependence, meaning cells like to be anchored to something. They don't just like free floating around. So oftentimes they won't divide if they aren't attached to something. So those are just some of the possible regulatory factors in cell signaling associated with the cell cycle. All right, you made it.